So, good morning, everyone. Today, here we are with a new chapter that is Principles of Inheritance and Variation. More commonly among students, it's famous in the form of genetics. So, this chapter is very important because it's one of the chapters which makes interest in the minds of students as this involves a lot of concepts, little bit of mathematics, and more of conceptual understanding than rote learning. So the questions of this chapter are also very interesting to solve. So I hope you all will have a very good journey while solving the questions with me. So with this, let us begin with our question solving for the understand the subsequent concepts. So with that, let's begin with the first question, that is experimental
Shubh,
verification
Shubh,
of
Shubh,
the chromosomal theory of.
Shubh,
Yes, sir.
uh, please check your
Yes,
WhatsApp
sir.
regularly. Please check.
Okay. Oh, okay, sir. Voice is breaking. Now also, sir.
Can we communicate over WhatsApp?
Experimental verification of the chromosomal theory of inheritance was done by, there are four options: Sutton, Boveri, Morgan, and Mendel. Choose the correct option and be a bit careful while choosing. Okay. So, if we move on to the correct answer, it is option C. That is, experimental verification of the chromosomal theory of inheritance was done by Morgan. See, let me explain the thing first. Okay. That is, you have heard the names Sutton, Boveri, and Morgan. All three have heard under chromosomal theory of inheritance. Okay. While the laws of inheritance that were given, the law of dominance, segregation, and independent assortment, they were given by Mendel. Correct? This much we know. That the Mendelian theory of inheritance, laws of Mendel, when we come to the chromosomal theory of inheritance, it was first proposed by Sutton and Boveri. Okay, they first just proposed the theory. Okay, it was not that they made the theory or they proved it. But when it comes down to prove the chromosomal theory of inheritance on ground with an experiment, that was done by Morgan. Okay, very well. Okay, because these people tend to forget. Okay, so what is the chromosomal theory of inheritance in one word? If I have to say, that is simply when you go from cell division from G1 phase to the formation of the germ cells. Okay, I'm taking an example of a meiosis. Okay, what happens? See, I'm drawing the chromosomal theory of inheritance. I'm explaining via a drawing. Okay, try to understand this. Okay, because if you don't understand this now, then the experiment of Morgan is understanding is even difficult. Okay, see, first, if we are beginning from G1 phase, what do we have in G1? For example, in G1, we have this. Okay, for example, let me just think in G1, we have like this. Okay, we now go forward in G2. One second, just now you go forward in G2. So what happens in G2? What do we get in G2? It will turn into a bivalent, right? Not actually a bivalent, I must say, but then the genetic material duplicates because what happens in S phase? In S phase, the DNA material replicates, right? So what I will get, what will I get in G2? I will get double of this thing. This also doubled and this also doubled. Okay. I should actually have made them with different colors, But then, chalo, for your benefit, I will writing 1 and 2. Okay. This will be 1, 1 and this will be 2, 2. Because the materials are identical, right now. Or let me just do it like this: 1, 2, 3, 4. What will happen? This will be 3 ka replicate. This will be 4 ka replicate. Simple. Now what will I have? I will have the anaphase of meiosis 1, right? Where the first segregation will happen. I will only take the major events. Okay, I'm not taking all events. I will now enter meiosis. What will happen? Prophase. Again, there will be some condensation. There will be etc. Many things will happen. Metaphase, again, many things will happen. There will be metaphasic plate, and there will be arrangement of the chromosomes on the equatorial plate, and many things. In the anaphase, the main event, what will happen? One main thing. That is, there will be the chromosomes. Sorry, there will be the chromosomes. Okay, and this time, what will happen? They are in a bivalent form. So is this correct? This is not correct. What is the correct form of drawing them? And this. Now do you think I have drawn correctly? Yes. So now how will I represent it? If I have to represent it, what will happen? So you can see the material is assorting itself in a very independent way. Okay. In short, what is the thing to get it down? What was there in G1, I will get it all back in the germ cell stage. I will get it all back in the germ cell stage. The same thing I will get back in germ cell stage. In short, okay. Because there are questions regarding this now in front, so I will be explaining it in better way and over there. As of now, I will be getting the same thing back in the germ cell stage. You should understand this much. What was there in the G1 phase in the parent cell? The same thing I will get back in the germ cell stage. This is the major takeaway from the entire chromosomal theory of inheritance. Okay, they state a statement actually. Okay, I'll read the statement first, then I'll tell you. Okay, or I'll just tell you the statement first. Okay, in my words, I'm telling. Okay, just try to understand this thing. Okay, they told one very simple thing. Okay, that is chromosomes behave just in the same way as genes. Genes assort them independently. Similarly, chromosomes assort them independently. Suppose here, chromosome 1 and 3, I mean 1 and 3, which are together forming the bivalent, they won't be going away anywhere. They will be staying together. And that is independent of the other set. As simple as that. This much if you are clear, you will be there enough to understand the chromosomal theory of inheritance. Okay. Now, if I move forward to the next question, the contrasting pair of factors in Mendelian process are called. Read the words properly. Okay, contrasting pair of alleles. Sorry, contrasting pair of factors in Mendelian process are called what? Let us see who gives the correct answer. Okay. Here also I will discuss a little bit about the Mendel's laws and all. Okay, so that things are a little bit more clear to you and so try to answer this. Four options are there: multiple alleles, allelomorphs, alloloci, and paramorphs. So if you move on to the correct answer, it is option B, that is allelomorphs. Okay, control different expression of traits to the same character. See, simple logic. Contrasting pair of factors. How are we contrasting it? On the basis of the morphology. And we know that different loci in the same gene, or what we can say is that in like a same chromosomal part, if we are having different loci of the same trait. So what will happen? That those are known as alleles. So alleles having different morphologies, they are known as allelomorphs. As simple as that. Okay. So what are those? What are these contrasting pair of factors? What are some allelomorphs? Let us just see. Okay. See, Mendel, when you're reading Mendel's theory, always remember the number 7 is very important for Mendel. Mendel has, I think so, number 7 is very lucky for Mendel. I don't know what is the thing, but it is so. Just let us see why. See, Mendel took some characters and he set some dominant traits for that, and he had set some recessive traits for that. Okay, that's it. What are the traits? First, he took seed shape. He made the proper round shape for dominant, and he took a wrinkle shape for, and yes, and he took a wrinkle shape for the recessive one. Okay. Then he took seed color, where he took again yellow color to be for the dominant trait, and he took green color for the recessive trait. Then he took the pod shape, where he took the full pod, or like the entire pod, swollen pod, as the proper pod shape, while the constricted pod, what is the constricted pod? It is like this. Okay. Then what did he take? He took pod color. And the pod color, he took green pod as the, like he took green pod as the dominant one, and he took yellow pod as the constricted one. Okay. Then what he did? He took the flower position. Where he took, like in the entire plant, the axial position. Okay, not sorry, axial position. No, I drew it incorrectly. He took the axial position. Okay, this is the axial position. Like it won't be at the top; it will be on the sides. In the axis, it will be there. And if it's a terminal flower, like whatever branch you go, it will be on the terminal parts only. It is terminal here. Okay. Followed by this, what else is there? There is stem height. This is also 1 point. So stem height, what will be this? This will be tall here, and this will be dwarf here. Okay. So these are the 7 characteristics that Mendel took in consideration. Okay. While he was taking the pair of factors that he will study on. Okay. So this is the thing. Okay. Fine. See, why am I telling everything is about 7 in Mendel? Mendel was born on 7th of July, I think. Okay. Then he worked for 7 years. He wrote 7, he took 7 traits. Okay. Even I think he died in some 7 or something, maybe. There was this thing. Okay. So there were like many things about Mendel in 7. Okay. So that is the thing. Next, if we are coming forward to the next question. I hope this is clear for you, right? Okay. Very interesting question. A man of A blood group marries a woman of AB blood group. Which type of progeny would indicate the man is heterozygous? Option A, AB blood group. Option B, A blood group. Option C, O blood group. Option D, B blood group. Okay. Meanwhile, you try solving the question. I will explain on the side what Mendelian inheritance actually looks like. Okay. Little bit I will explain. Okay. So that you will at least get an understanding of how the thing goes. Okay. See, for example, now how do we write a dominant trait? You all know these things, right? For example, I'm taking the example of tall and dwarf. So tall I will write with capital T, dwarf I will write with small t. But dominant I will write always capital letters. That is like capital T, and whichever is the recessive trait, that I always write with the small letter. That is what? The small t. Now, how do we do a dominant trait? Dominant trait can express individually, and so much so that it overcomes the recessive one. And one dominant trait in a, because we are all diploid organisms, no, so we won't be expressing a single t. We will be expressing either a single t, small t, or we will be expressing capital T, or we will be expressing small t, or we will be expressing small t, capital T. We will be expressing what? You will be expressing a pair of two alleles because we are diploid organisms, right? This you have to understand, number one. Okay. Now, what is the major important thing over here? You understood dominant how we write, recessive how we write, and how dominant expresses individually overcomes recessive. Next, recessive. It to express itself needs both its alleles, which means only small t will be dwarf. Rest capital T, small t, and capital T, small t, all will be tall. This you understand from this thing, right? Because here the dominant trait what does it overcomes the recessive one. That is the basic thing, right? So this is the Mendelian trait, right? How do we do a Mendelian cross? I'm just explaining this in short, okay, so that you have a basic idea of how do we do a Mendelian cross. For example, I have a tall plant and I have a short plant. I want to do a cross of them. Okay. This is the F0 generation. So when I come to the F1 generation, what will I get? Let us draw a cross. What I get is all plants of this thing. This becomes my F1 generation. Now, if I do a F1 cross, that is, now if I cross a plant of F1 generation, what will I get? I am getting many sorts of plants, right? Where what is in ratio of 1 is to 2 is to 1. But if I take the ratio of tallest to short, it will be what? It will be 1 plus 2. That is 3 is to 1. This you all know, right? This you all know, right? That how we do a Mendelian cross. You all know this thing, right? I expect that you all know this thing. Okay. But what happens if I take the trait of blood group that is given in the question? How does this express now? This is an exception. An exception known as incomplete dominance. Because what happens in blood group has three alleles. One is IA, one is IB, and one is IO. IO is recessive, while IA and IB both are dominant. That means if IA is there, it is A blood group. If IAIO is there, it is A. IBIO, it's B. IBIB, it's B. But IOIO, it's O. But what if IA and IB are there? It will be AB blood group. And this characteristic is known as co-dominance. Is this thing clear to you? What happened in the blood group thing? If this thing is clear to you, take a minute to look at it. In codominance, if there are more than one dominant alleles, in the pair of alleles, both the alleles express themselves together, independent of each other as a whole. No dominant allele takes over the other. Both express individually, both characteristics are present, no characteristic is suppressed. This is incomplete dominance, seen in blood group antigens. This thing is clear to you? Let us move forward to the question. Question says, the man is of A blood group. What does A blood group mean? It can be IA, it can be IAIO. Marries a woman of AB blood group. Now the lady is of AB blood group. That means the lady is of IAIB. Confirm. Which type of progeny would indicate the man is heterozygous A? A question has said me कि I have to prove that the man is heterozygous A. And this they are indicating me by showing the progeny. Okay, let us see. First, let us assume the man is heterozygous A. So let us make a cross between these two. So what will I make? I will make IAIO by IAIB. I will make cross, right? So let's make a cross. This is IAIA, IAIO, IAIB, IBIO. Now you think, these two will I get in homozygous? These two will I get in homozygous? I will not get, right? Yes, I will not get these two, right? If I'm having one, if the man is heterozygous A, if if the man is homozygous A, will I get this IO progenies? I will not, right? And what does this indicate? That either A or either B can be the answer. Now you think, if it is a home, both cannot be the answer. It will be a single correct option. So, which option do we need to eliminate? Let us make the homozygous option and let us see. IAIA, IAIB. You will soon understand the difference. See, IAIA. And you see A blood group also comes in homozygous. It also comes in heterozygous. So, which is eliminated? A blood group is eliminated. The answer is only B blood group. Check for the answer. It's only option number D, that is B blood group. So, our answer turns out to be option number B. Option number A is eliminated. Okay. So, you all get the solution, right? This is pure maths. Pure maths. Okay. You have to be a little bit attentive while solving genetics questions. You might know the solution, still you might make a mistake. This is the beauty of genetics. From my experience, I'm going to share one thing with you. You know where I got the best genetics questions to solve in a live exam? That is in KVPY. There is a KVPY SX paper, the paper for class 12. That paper had beautiful questions of genetics combined with biotechnology. Like those were most probably one of the most beautiful questions. Okay. Then I think you might have also heard about the biology Olympiads. Over there, one of the best signature genetic questions come. Okay. Like, I mean, that were the most golden genetic questions I've ever seen in my life. Okay. Like such beautiful integration of concepts with anything. So genetics can be made very beautifully. But for that, what you need? A beautiful understanding. Okay. So with that, let's move on to the next question. TT mates with like small t mates with capital T small t. What will be the characteristic of offspring? Very easy to answer. Still, you all try to answer this. I'm showing the cross in the adjacent slide. Capital T small t cross small t small t. So what does it come out to be? It comes out to be 50% recessive, and it comes out to be 50% dominant. So correct answer comes out to be option number B, right? This is, I guess, a very easy piece of cake question. You'll understand like how 50% recessive thing comes. Okay, this has nothing much for me to explain, right? This you all can understand easily by your own understandings itself. So clear with this? Okay. Among the following characters, which one was not considered by Mendel in his experiments on pea? Trichomes, glandular or non-glandular, seed, green or yellow, pod, inflated or constricted, stem, tall or dwarf. I explained it to you under the heading allelomorphs. Try to answer this now. See, you all understand Mendel's laws, right? Mendel's theory of inheritance, whatever he has stated. I'm still explaining them a little bit to you. Okay. Just listen to me. Okay. See, Mendel did what? Mendel had some theories on genetics. Okay. He proposed these ideas to the then that was the era of Darwin. Okay. When Mendel was working, no, there were many scientists working at that time. And the majority of scientists at that time were working on the evolution patterns. Okay. So that is why Mendel's ideas were like they sidelined. Mendel was saying that people evolve different traits because of the genetic makeup. But people were like, no, it's evolution, it's natural selection, it's genetic. They were not ready to believe the most popular guy in that era. He was like there, okay. And then there were some British scientists like Haeckel and all over there, okay. We're all working on evolutionary patterns and everything. Even the scientists who are botanists started to working on, matlab, on all the animal evolutions. Okay, they started working on the evolution of animals and everything. So that was such a big era of evolution. And that is why you know what happened. Mendel proposed a theory of genetics without doing all these experiments on pea plant. But that experiment was rejected. That theory was rejected. They were like, no, everything is evolution. Everything is natural selection. I will not accept this theory of genetics. You don't have any base to prove it. Seriously, Mendel didn't have any base to prove it. Mendel also put the theory on pea plants and everything. Okay. At that time also nobody gave recognition to it. Nothing. Okay. Mendel made the experiments, Mendel wrote them, and Mendel died. After Mendel died, the microscope was invented. In the microscope, they saw all these things. They saw nucleus, they saw chromosomes, they saw genes. And then they were like, okay, some guy some few years ago told us about all these things. At that time, we didn't believe him. But then he was right. And then they realized their mistake that yes, they were right. And one guy actually told the correct thing. Okay, things evolved from genetic materials. Genetic makeup of the organism is important. And thus, there came the pioneer of molecular biology, late Gregor Mendel, not Gregor Mendel. Okay, and that is the beauty of molecular sciences, that it was discovered by a scientist, but his ideas were not approved when he was living. His ideas were approved by other people some 10, 15 years down the line when he died. Okay, that is why you will see Mendel died in this year, and after 10 to 15 years, his ideas are published. Okay, so this is actually the thing. Okay, this is very contrasting. This is sad, but this is true. Okay, so this is the story of Mendel. Okay, let's come back. So which is not considered by Mendel in his experiments. Let us see what are the traits. See, these are the traits: seed shape, seed color, pod shape, pod color, flower position, and stem height. Okay. Now out of this, which one do you feel is the one over here? It is trichomes, obviously. So not at all a big deal. You can all understand what is the thing. Okay. Next, another beautiful question. Okay, let us solve. How many different types of genetically different gametes will be produced by a heterozygous plant having the genotype capital A, small b, capital B, small b, capital C, small c? I will also give you a trick of solving these questions. Okay. The correct answer is 4. Okay. I am telling this first only. How to solve these types of questions? Okay. See, if I draw a very big thing like this, suppose capital A, small b, capital A, capital B, small b, capital C, small d, capital E, capital F, small f. I am writing this bigger thing. I want to see how many different types of genetically different gametes have been produced from this. Shortcut technique. Listen to this very properly. How many pairs of heterozygous gametes are there founded? One pair, two pair, three pair, four pairs. So four pairs of heterozygous gametes are here. So number of different gametes will be 2 to the power 4, that is 16. So if there are n number of heterozygous pairs, that will indicate 2 to the power n different gametes. Is this thing clear to you? This I'll make in a box. This becomes very clear to you. What is happening? Hmm. This you should never forget. If there are n number of heterozygous pairs, there will be 2 to the power n number of different gametes. Keep this thing very straight and very clear. You should never forget this. You can forget anything else, but you cannot forget this. So in capital A, small b, capital B, small c, capital C, right? Small c, capital C. So in this, you can see that there is one, there is one. So there is two pairs. That means there is 2 to the power 2. What is the correct one? Did you get the correct answer now? What is the answer? Is this now clear to all of you? If it is so, then we'll move on to the next question. Okay. That is, next question is question number 7. That is, radioactive thymidine, when added to the medium supporting living mammalian cells, gets incorporated into the newly synthesized DNA. Which of the following types of chromatin is expected to become radioactive is if the cells are exposed to the radioactive thymine as soon as they enter the S phase. Is it heterochromatin, euchromatin, both hetero and euchromatin, neither heterochromatin nor euchromatin, but only the nucleolus? Try answering this question. This question is actually not for genetics. This is a little bit common sense question. Try to answer it. You will be able to answer this. This is not a very rocket science question. Okay. See, giving you some hints. They entered into S phase. They have mentioned they are inserting radioactive thymidine in living mammalian cells, where it is incorporated into newly synthesized DNA. They are asking you only this thing, which type of chromatin is going to become radioactive. So you have to think the type of chromatin and what exists in the S phase, and that will be your answer. As simple as that. So if we move on to the correct answer, it will be option number B, that is heterochromatin. Because you have to understand that chromosomes become condensed until metaphase. Okay. So it will be heterochromatin at that time. Okay. Heterochromatin is what? It is a very tightly coiled and stains darkly in interface. So if you are adding radioactive thymidine, it will go and it will bind to the newly synthesized DNA fragments in the heterochromatin present in the S phase, not anywhere else. Okay. And what is happening? See, so sorry. I was explaining about heterochromatin, but then the answer is euchromatin. See. Let me draw and let me explain. My bad. See, first of all, what is euchromatin? Euchromatin is that chromatin which is less coiled, right? What is heterochromatin? That is tightly coiled. Now, when the cell is in S phase, what is happening? There is DNA replication going on. Now, for DNA replication, what needs to happen? The chromosomes need to be uncoiled. Now you have given radioactive thymidine. So it will bind to euchromatin. Is it now clear to you what happens? That is the answer. Radioactive thymidine here binds to the euchromatin. Okay. Next, we have a fruit fly, heterozygous for sex-linked genes. Okay. When mated with a normal female fruit fly, the male specific chromosomes will enter egg cells in the proportion of, see, here. First, you all try to answer, then I'll explain you with the correct solution. See, this comes under the portion of X-linked inheritance. Okay. How does this go? See, as per me, the answer should be 1:1. That is the correct answer. But how does it come? See, they have taken a normal female fruit fly. Female fruit fly is XX. Male fruit fly is XY in the gametes. Now, what they have taken is they have taken the female gamete which only has X. But what can male gametes have? Male gametes can have X or male gametes can have Y. So they have asked me, when it is mated to the normal female fruit fly, the male specific chromosomes will enter egg cells in the proportion of. Okay, in the egg cell there is only the X. But now the let's take it as sperms only. They have either X or they have a Y. Now what is the probability of them getting into the X? Here it's 50%. Here it's 50%. Hence the ratio is 1:1. As simple as that. We know that they enter independently. I told you no. Chromosomal theory of inheritance. What does it state? That chromosomes segregate in the same order as the genes do. That is the theory. Hence, both of them will have equal probability to come and meet together. And thus the answer is 1:1. Any doubt in this? You see, male specific chromosome will have half as much chance as the other to enter the X cell, and hence the proportion will be 1:1. Okay. Moving forward. After crossing between two plants, the progenies are found to be male sterile. This phenomena is found to be maternally inherited and due to some genes which are present in. I'm giving you one hint. This is also known as cytoplasmic inheritance. Okay, so now try to answer this. After crossing between two plants, the progenies are found to be male sterile. Phenomena is maternally inherited due to the genes present in. The correct answer is option C. I gave you the hint cytoplasmic inheritance to confuse you guys. Option D is not the correct answer. It is known as mitochondrial inheritance or cytoplasmic inheritance. Why cytoplasmic inheritance? Now understand. See what happens. There are some the mitochondria, has some genes. Like you all have studied this in cell, right? See, very well. Understand these things. See, if you understand, no, it will be very easy for you to comprehend and apply it. Okay, when you're needing to apply, try getting what I'm trying to say to you. Yeah, you have studied this in cell that mitochondria and chloroplast, both of them have their own DNA. You have studied this, right? They have their own DNA. On our own DNA, right? It has own genetic material only. Or it is a prokaryotic material. No? They have their own genetic makeup. Now, only mitochondria is present in humans. So that also contains their own genetic material. So that genetic material of mitochondria gets inherited, but only of the female gamete, not the male one. Why? Because its nucleus is only taken during fertilization. Okay, that's what happens. This gene from mitochondria only comes from female. Hence, it's called mitochondrial inheritance or cytoplasmic inheritance. Why cytoplasm? Because mitochondria is in cytoplasm. It is not a part of the nucleus, right? So it is cytoplasmic inheritance. Okay. Now, there are many examples of mitochondrial inheritance which you need not understand at this moment. But then understand this much, that all characters of the mitochondrial inheritance present in the mother will also come to the child. Okay. There are many types of disorders. Okay, so there is many mitochondrial disorders. For example, any enzyme deficiency or anything. Okay, if that exists in the mother, no, mitochondrial inheritance. Did you understand? So this is all about mitochondrial inheritance. Okay, so moving forward towards question number 10, we have alleles that produce independent effects in their heterozygous conditions are. We have already discussed this under AB blood grouping. Try to link codominant alleles, epistatic alleles, complementary alleles, supplementary alleles. So moving on, the correct answer is epistasis. Epistasis. No, epistasis is not the correct answer. They are the explanations. Option A is the correct answer. That is codominant allele. What is codominance? I told you that when two dominant alleles express themselves individually, as in IA and IB. What did these two do? They together express them as AB blood group, right? And that is what you need to be very careful about. Okay. You want to read about the other alleles? Let us read. What is epistasis? Suppression of the normal phenotypic expression by a non-allelic gene. Suppose two genes make up the allele. There is a third gene. Third gene is doing nothing. It has nothing to do with that allele. For example, there is an allele for tallness and dwarfness. Another allele does something for color. But when that is present, it suppresses the tall gene. That is known as epistasis, epistatic gene. Complementary are two genes giving a synergistic effect. For example, I write something as like T1 and T2. When these two are present, it becomes tall, more tall. Only T1 is present with T, it becomes tall but little bit tall. When T2 with T, it also becomes the same amount of tall as T1. But when both the tall alleles are present, it will become extra tall. That is known as complementary effect. Supplementary genes are again what? They are also doing like this, but then they are present in different gene loci, different traits. These are same traits and different gene loci. Okay, these are some of the extra knowledge that you might get. So this I will like you to take a screenshot so that you can refer to this later on. What is epistasis? What is complementary gene? What is supplementary gene? This can be asked to you in a matrix match question at maximum. So you should know only the what is it. You should not go into depth as to what is the ratio and all. There are many things, but then I would suggest only remember the definitions. Okay. Next question. When two genetic loci produce identical phenotypes in cis and trans position, it is considered to be. That's a good question. Try to answer this. This is theoretical. You just need to know the name, but then at least you should know. And the correct answer is option B. That is different genes. Okay. See why. Try to get this thing. This is actually known as a test known as cis trans test. Okay, also known as complementation test. This is not given in NCRT. This is like a topic that we study under MBBS. Okay, in MBBS you have to study genetics under anatomy. Over there we study all these things. Okay, so what is complementation test? It is nothing but just to check for mutations. Okay, whether suppose some phenotypic change is there due to a mutation. So whether it is an allelic variation or whether it's like it's a monoallelic variation or it's a biallelic variation. Just to check that we do this thing. Okay, this much you know, and this is also discovered by Edward B. Lewis. You should know this much. Okay, you can read the rest of the explanation, but then what I want you to know is just this much. Complementation test, also known as cis-trans test. What is it? To check whether two mutations with a specific phenotype represents different forms of the same gene or are variations of two different genes. This much. Okay. What is the test? You might know, you might not know. That no one will ask. And they might ask you the name of the American geneticist who discovered this. That is Edward B. Lewis, who said if the combination of two genomes containing different recessive mutations yields a mutant phenotype, then there are three possibilities where one mutation affects the expression of the other. Okay. This much if you're understanding, it's enough. Okay. Okay. Enough for this. We can move on to the next question. In a dihybrid cross, capital A, capital B cross, small a, small b. You have been asked the F2 progeny of capital A, small b, capital B, capital B, capital B, small b, and capital A, small a, capital B, and capital A, small a, capital B, small b occurs in the ratio of what they've asked you. So now you have to write the proper thing. So how will you write? I'm giving you a shortcut for this. Okay. Try to get this shortcut. This will be easier for you to remember. Okay. Whenever something like this comes, no. First of all, see what are the things. When we are doing capital A and small a. I will get like just wait for a minute. Yes. So here, what are the things that can happen? Either it will be capital A, small a, or it will be small a. Also, there for the B, what will be there? There will be capital E, small b, and small b, right? These are the main combinations that can be formed. So let us make a matrix 3 into 3. Okay. Genetics, is very interesting to do, but this trick will be something new for all of you. This I learned after coming in MBBS. Okay. One of our teachers taught this. This is very interesting technique. Just learn. See how to find out the ratios. Right? Like this capital A, small a. Again, right. Capital A, small a. Capital A, written. Now what to do? Now change the order and write capital B, capital B, small b. Capital B, capital B, capital B, small b. Capital B, capital B, small b. Now write 1 2 1, 2 4 2, 1 2 1. Now find out the ratios. Capital A, capital B is this one, right? It is this one. Capital A, capital B, small b. This is this one. Next, capital A, capital B. That is the capital A, small a, right? And then two capitals. That is this one. Next, what? Capital A, small a, this one. The ratio is what? 1 is to 2 is to 2 is to 4. How is this technique? You all loved it? This is one of the best techniques. I'm telling you. Solving these questions was literally very monotonous, very tiring, very lengthy question when I used to solve them in my UG days. Okay, I mean before NEET. But then when I came to college and our anatomy professor taught us this on the board, I was like, is this how can we even do this? Why did I waste so much of time? Now see the techniques. Okay. So that's what you learn after coming to medical schools. And you are learning this from me. So I hope this helped you a lot during your exams. Okay, coming up to the next question. This is again easy question. Which of the following characters represent inheritance of blood groups in humans? Dominance, codominance, multiple allele, incomplete dominance, polygenic inheritance. Let me write some hints for you so that you can answer it very easily. There is IA, IB, IO. Out of this three, any two are inherited, and but if IA, IB is inherited, you get AB. So now answer this. Why have I drawn this? I'll explain you. But if I get IAIO, it will give me A, not AO. And if I get IBIO, it will give me B and not BO. Only IOIO only will give me O. Now answer this question. What I
Shubh,
see.
Shubh,
You have chosen the correct option. If you choose option B,
hello, Shubh.
yes, sir.
Shubh, you need
Yes, sir.
to log out and log in again, as I said earlier, because your network is not stable and voice is continuously breaking. Well, today's session is matlab, like what I say. Anyhow, log out and log in again.
Okay, sir. Okay, sir. I'll just log out. Sir, now the voice is better, sir.
Please start with previous question.
Sir. I'll sir go like from question number 11, sir. Like this question I was explaining, previous question, sir, from I'll start.
Yes.
Okay, sir. Okay. So we have question number 12 in front of us, that in a dihybrid cross, where we have been given capital A, capital B, cross small a, small b, small b. In an F2 progeny, we have been asked for the ratio of the four genes. Okay. In which ratio does it occur? So here I have a trick for you to solve. Okay. What is the trick? Let us see. Just hold on for 1 second so that I can explain it to you in a better way. Yes. See over here. How to solve these questions? First of all, write this like this: capital A, capital A, small a, and small a, small a. Okay. In a 3 into 3 matrix, you have to write. This is a shortcut technique to solve this question. Capital A, capital A, small a, small a, small a. Write the same thing in rows for the A allele. Then now for the B allele, what to do? Write the same thing in vertical rows. As in capital B, capital B, small b, small b. Write them in vertical rows. Then now compare them as to 1 is to 2 is to 1, 2, 4, 2, and 1 to 1. You know, right? For a monohybrid cross, the ratio is 1 is to 2 is to 1. So if I multiply it with 2 and I write in the middle, I'll get the proper thing. I'm basically adding these two to get this. Just get it like this. Okay, as simple as that. Okay, so now if I make this entire grid, okay, if you understood what to make the grid, then any ratio you can find. What is the ratio now? See, we have been asked for capital A, capital B, capital B. That is this one. That is this one they're asking for first. Next, they're asking us for capital A, small b, capital B, small b. Next, they're asking for capital A, small a, capital B, and next they're asking for this one. That is capital A, small a, capital B, small. So if we see the ratio, how does it come out? 1 is to 2 is to 2 is to 4. Hence the answer is option number D. You all get this thing right? This is one of the techniques that I learned after coming to medical school. Our anatomy professor taught this. So when I used to solve this question during my NEET days, this used to be a very tough question. But after coming to MBBS, it became a piece of cake. So I hope this trick also comes in handy for all of you while solving the questions. Okay. So with this, let's move on to the next question. That is, which of the following characteristics represent inheritance of blood groups in humans? There are five characteristics written. Okay. Dominance, codominance, multiple allele, incomplete dominance, and polygenic inheritance. You can pause to choose the correct answer. So the correct answer comes out to be option number B. That is A, B, and C. That is dominance, codominance, and multiple allele. See why? First of all, it's dominance. Why? IAIO is giving you A blood group. It's not giving you A and O both. A. Why? Because O is a recessive allele, A is a dominant allele. As simple as that. Okay. Out of these A, B, and O, the main three alleles for blood groups, right? Only A and B are dominant. IO is a recessive allele. Hence, there is a characteristic dominance as well as recessiveness. But there are two dominant alleles. And when those two dominant alleles come together, like IA, IB, they express them together as AB. This is known as co-dominance. Okay. That you can see. Also, how many alleles are there? There are more than two alleles. Hence, we can call this as multiple allelism. And hence, there are multiple alleles also. Now, due to the presence of dominance, codominance, and multiple allelism, we give the answer as option number B. Is this thing clear for all of you? Okay. So, with this, let us move on. A child of blood group O cannot have parents of blood groups. Which you think? I have IOIO. If I have one recessive allele, what I cannot prepare is codominant allele. Can you imagine this in your mind? I can have I something, and I have IOIO. Now something will come from here, and from here IO will come. This something can be IAIO, it can be IBIO, it can be IOIO, it can be A, it can be B, it can be O, but it can never be AB. Now, can you imagine something out of it as to why it cannot be AB? And hence, what turns out is that our answer is option number A. Okay. Income. See, codominance does not mean that there won't be any recessive allele. There will be recessive allele, but the dominant alleles will not compete between each other. They will exist together independently. It's like two gold medalists having two individual gold medals. Okay, this was an example given to me. So I'm just sharing it with all of you. Next question. This is an easy question to understand. Mendel studied inheritance of 7 pairs of traits in P, which can have 21 possible combinations. If you are told that in one of these combinations, independent assortment is not observed, your reaction will be option A. Independent assortment may be wrong. Option B. Mendel might not have read all the combinations. C. It is impossible. And D. Later studies may be wrong. Okay. So now what is your opinion on this? Choose the correct option. So moving on to the correct answer. That is option number B. That is Mendel might not have read all the combinations. What does law of independent assortment state? That two different traits having that alleles, they will always express independently. Okay. When you are saying independent assortment is not observed, that means there are more combinations to study. You study all the combinations first. You will find a correlation between them. Okay. It is something just like that you found a ratio and you found that it is going irregular at one point. And you have just stopped doing it. No. You study everything, finish everything, then you see what is there, what is not. Okay. This is something which you have to get via imagination. Okay. Let me give you an example. For example, you saw a dihybrid cross. 16 combinations come out. I'm telling you 15 came out. What does that mean? I didn't study one combination, right? But does that make that study invalid? No, right? The same thing happens here. I hope you got my point, how this thing is going on. Okay? That is the thing. I missed one observation does not mean that that observation does not exist. It exists. But I have to work for it instead of saying it's invalid. Okay? Always remember this one point. This is very valuable. Okay? See, law of independent assortment is applicable to only those factors of genes which are located on different chromosomes. Probably the characters were present on the same chromosome and showed linkage, which Mendel might not have studied. What is linkage? We'll see in some upcoming questions. Now, a polygenic inheritance in human beings is, see, you need not study polygenic inheritance. You need to know the examples of polygenic inheritance. Because polygenic inheritance is not that deeply in your syllabus. You know the names mainly. So, examples of polygenic inheritance is, let us see the correct answer. This skin color. What is polygenic inheritance exactly? Is that different genes are there. It depends on how many genes are present, the effect will be additive. Suppose I'm telling three genes are there which code for skin color, A, B, and C. If only A gene is present, your skin color will be a little bit dark. If both A and B are present, your skin color will be darker. It will be darkest when A, B, C, all three are present. If all three are absent, your skin color will be very white. So what is happening? There is an additive effect of the genes. And my students, this is what polygenic inheritance is. I hope I made sense to you while saying what is polygenic inheritance. So that is the main essence. Next is two dominant non-allelic genes are 50 map units apart. The linkage is cis type, trans type, complete, or absent. We will study linkage in the upcoming set of questions. So for now, I just want you to understand what is map unit. Map unit is when we are plotting genes on a paper. We take the SI unit of gene as map unit. Okay, remember this. This is the SI unit of gene distance. So if two dominant and non-allelic genes are this much apart, what is this to be said? Let us see the correct answer. It is option number D, that is absent or incomplete. Obviously, 50 map units is a very long distance. If it was 1 or 2 units apart, then we could have said linkage might have existed. But since it is so far away, linkage is not there. This much we are rest assured of. Okay. So this is the solution for this question. That is, the linkage is incomplete. Okay. So with this, I will keep up till here. The next day, concepts like linkage and all the Mendelian disorders are left. We have covered for the day Mendelian inheritance, the monohybrid dihybrid process, the chromosomal theory of inheritance, and questions on exceptions of Mendelian inheritance. And we have also just touched upon the X-linked inheritance, which is again a part of chromosomal theory of inheritance. So we are left with the second half of the chapter of genetics, whose questions we will solve in the next session. Okay, so that is it for today. Revise NCRT, solve more questions of genetics to get your base stronger, and keep studying. Thank you, students.